And what we're going to do is what we're going to start with is just a very brief overview of what translation is all about. Now, at any given moment, any cell in your body is typically synthesizing thousands of proteins. Now, these proteins, after being synthesized, are transported to various locales or regions within a cell where they are used for various things. Now, in eukaryotes specifically, in eukaryotic cells, the synthesis of these proteins that are used so widely in the cell are actually guided by the process of translation. Now, by definition, translation is the process in which or by which the RNA that is produced during transcription is read or decoded by a ribosome to produce a specific sequence of amino acids. So, translation can therefore in that way be thought of as the mRNA-guided synthesis of proteins. And this synthesis of proteins is also ultimately indirectly guided by the specific nucleotide sequence or the DNA that codes for the mRNA that is produced during transcription. So here we see ourselves returning to the central dogma in which we have a gene in the form of a nucleotide sequence in DNA coding for a messenger, which is mRNA, which then proceeds to code for a protein. So that in a nutshell is the definition of translation. Now, what are the key molecular components of translation? The most important ones that you need to be familiar with will be messenger RNA or mRNA, which is the transcript, the messenger transcript, the transcript of DNA in the form of RNA that actually carries the genetic message. And this message is what specifies a specific series or sequence of nucleotides along an mRNA molecule. Now, the second important molecular component you need to familiarize yourself with is obviously tRNA or transfer RNA. And what tRNA does, it transfers amino acids from the cytoplasm, from the cytoplasmic pool of amino acids, and it takes those amino acids to a growing polypeptide chain that is being synthesized in a ribosome. Now, typically, any one cell will keep itself stocked with all the 20 different kinds of amino acids, and it will do so either by synthesizing them or by taking other compounds or making them from other compounds, or by taking them from taking them up from the surrounding solution. Now, the third important component of translation that you need to commit to memory will be the ribosome. Now, the ribosome is actually a structure that is made up of proteins and RNAs. And what the ribosome does is that it adds each new amino acid that's brought to it by transfer RNA to what is a growing polypeptide chain, resulting in the formation of a polypeptide or a protein. So, now before delving into the key mechanistic steps of translation, it's important for us to understand the actual code or the pattern that is actually used to read or decode genetic information. This is what we refer to as the genetic code, and that's what we're briefly going to look at or build on in the next slide. So, what you should know is that there are 20 different amino acids which are used in protein synthesis. Now, in the process of translation, the specific order of nucleotides in messenger RNA or mRNA will determine the specific sequence of amino acid residues in a protein or a polypeptide chain. This means that the specific order of amino acid residues in the chain is determined by the exact order of nucleotides in messenger RNA. Now, take note of the fact that I'm using the phrase amino acid residues, and this reflects the fact that peptide bond formation between two amino acids results in the loss or the elimination of a water molecule. And as such, the residues left behind after elimination of this water molecule is what is actually included in the polypeptide chain. Now, this order of amino acid residues in the polypeptide chain is actually what determines what we call the primary level of structure in a protein. And if you go back to the lecture in which I talked about the levels of protein structure, this should be easily and right away apparent to you. Now, we know that the primary level of structure is what determines both the structure and the function of a protein. So With that knowledge, we know that the order of residues, the order of amino acid residues in a chain is very important and actually very definitive in determining the functioning of a protein. So, because the exact sequence of amino acid residues is so important and therefore very tightly controlled, it makes sense that there must be some sort of molecular mechanism that determines the order in which amino acids are added to a growing polypeptide chain during translation. So, we've seen that translation is the process by which the genetic instructions, the form of mRNA, are actually decoded to produce a protein. And we've seen that the order of amino acids in this protein that is produced is what determines its function. And one other thing you should already know is that there are only 4 different nucleotides that may be present in an mRNA chain at any one particular time. What this tells us is that translation cannot be accounted for by the direct one-to-one correspondence where one nucleotide codes for another amino acid. What this tells us is that if there are only 4 nucleotides in mRNA, 4 different nucleotides in mRNA, and these nucleotides are supposed to code for over 20 amino acids, then there must be some sort of system or code which uses these nucleotides in different combinations to code for these 20 amino acids. Now, this is in fact what we call the genetic code, more specifically what we call the triplet genetic code. So we're going to move on and talk about the genetic code